Hey guys, my name is Nikhil, and today we're going to talk about the innate immune system, specifically the complement system. So what is immunity? Well, immunity is your body's way of protecting you from dangerous germs and chemicals. So immunity is basically the thing in your body that's protecting you from the bad things outside. Where is immunity? Well, you can't really see immunity, but it's in your body, and you keep getting sick because of your immune system. It tells you that your immune system is working hard to protect you. So how does the body stop germs? Well, there's two ways to actually do it. One of them is called innate immunity, and that consists of policemen, right? So policemen can actually respond to most situations, and they can stop most germs. And so I like to think of these as the policemen. But what if the germ is a really nasty germ? Well, that's when we use the specific immunity, and that's when we need what I like to think of as the doctors. And these doctors take care of the individual germs, right? And so they can get rid of them. So before you really understand what your policemen are, you need to understand two different concepts. So one of them is a protein. Proteins are found in meat and beans, of course, but proteins are actually like really small buildings, right? So buildings can be different things, like factories, apartments, clinics, and many other things. And just like those buildings, your proteins can do many different things. What about your cells? Well, imagine that your body is made of little tiny Lego blocks. What if these Lego blocks were really small? Well, that's how your body actually is, and these little tiny blocks are called cells. So what is complement? Well, complement is a group of 30 proteins that is in your blood, and these proteins help us remove germs from our bodies, and that's what keeps us healthy all the time. So where is this complement made? Well, there's a part of your body called the liver, right? And the liver keeps making these proteins and it keeps sending it throughout your body so that you can fight any germ at any given time. So there's really many different ways of making this complement so that we can protect our body. One of the main methods is called the alternate pathway. So before we really get into that alternate pathway, let's discuss this very special protein. It's called C3. C3 is a special complement protein, and it's important because it's sticky and it can attach itself to germs, right? This is really important because that makes the body want to eat it and get it out of the body. So cells in your body will actually go and eat those germs. So when C3 actually gets into water, it turns into something called IC3. And when IC3 is in the blood, it can attach to something called factor B. And when the two meet, they can do some pretty cool things, right? So this thing on the side is called IC3B. So IC3B cannot really do anything because the factor B sitting on top of it is kind of lazy. And so what we use is something called factor D, right? And factor D is like, hey, I can help you here. So it actually cuts off a part of factor B, right? And that turns it into big B, little b. And big B, little b is not that lazy. And so IC3 big B, little b can actually do stuff. So IC3 big B, little b uses C3 proteins to make two new proteins. The two new proteins are called C3A and C3B. Remember that C3B is that sticky protein that we put on germs. Here's another word for you. It's called a convertase. We call it the C3 convertase because it converts C3 into C3A and C3B. So anything that converts C3 into C3A and C3B is called a convertase. The only issue is IC3 big B little b is kind of really slow at making C3B. So instead of making more IC3 big B little b, which would make everything slower, we make a new convertase called C3b big B little b. And we make it the same way. We take the C3b we made in the last slide, right? We use factor D, just like last time, and we cut off that lazy b, make it big B little b. And this one can convert C3 into C3A and C3B. And it's really fast. So we call it C3b big B little b. but it's so special and so good that we gave it a new name. We called it the alternate C3 convertase. So your body has a meal? Well, your body actually likes to eat things that have C3b on them, and there are special cells that the body uses to do that. These cells we call macrophages, right? And they especially enjoy eating things that are covered with C3b. The thing is, as soon as they eat them, they're like, oh no, we have intruders. And so what they do is they send out these messages that we like to call cytokines. Where do cytokines end up? Well, cytokines actually go back to your liver, right? And the liver's like, oh no, we have germs, we must protect ourselves. And so it makes new proteins. These new proteins are called mannose-binding lectin, MASP1, MASP2, and C-reactive protein. So that was the alternate pathway, right? Because we actually made a thing called the alternate C3 convertase. Well, let's look at two new pathways that this made. One is called the lectin pathway, and the other one is called the classical pathway. So the first thing that happened is that the lectin actually attaches to the germ. Think of this thing at the bottom of the slide as the germ. The lectin protein actually binds to it. And when it does that, MASP1 and MASP2 attach themselves to the lectin. So what does the lectin and the MASP1 and 2 do? Well, there's another protein called C4, and these can actually cut C4 into C4a and C4b. The cool thing is it can actually do another thing. It can cut another protein called C2 into C2a and C2b. So what about the other one, C-reactive protein? Well, C-reactive protein attaches to another protein called C1 that's always in the blood. And when it does that, it attaches to germ cells. CRP and C1 can actually do the same thing that lectin, MASP1, and MASP2 do. It can cut C4 into C4a and C4b. It can also convert C2 into C2a and C2b. So the C4b and the C2a that we made, they come together to form another convertase, right? For C3, to convert it into C3b and C3a, just like our alternate C3 convertase. We call this protein C4b-C2a. It's also given the name classical C3 convertase because it helps a lot. So in review, think of these three pathways as eating germs, right? Germs are like cookies without their chocolate chips. Yes, cookies are tasty, but if you think that the macrophages are cookie monsters, they'd really prefer that chocolate chip, right? So how do you get germs to look like chocolate chip cookies? Well, you stick some complement protein on them, right? That complement protein is C3b. And there's three ways to make C3b to stick on germs so that macrophages can eat them. One is the alternate pathway, where C3b big B little b is used to make it. One is the lectin pathway, where C4b-C2a is created to do so. And similarly in the classical pathway, you use C4b-C2a. So let's talk about the membrane attack complex, or the MAC. Macrophages can eat germs, but what if you really want to kill a germ, right? So these things will actually poke holes in the germ. Keep watching to see how. So we used the convertases we had from last time, and we're like, hey, you know what would make these really good? Let's stick another C3b on them, right? That'll make them really fancy, and it actually does. So these are given big names. These are called C3b, big B, little b, C3b, and C4b, C2a, C3b, right? So they can both cut this protein called C5, and it actually makes it into C5a and C5b. So C5b uses three more proteins, C6, C7, and C8. And C5b is like, hey guys, come on, let's do something cool together. They're like, okay. So when C5b, C6, C7, 8 come together, they attract this other protein called C9, and they make this huge circle right in the germ. And the germ's like, oh no, I got poked! And it actually deflates like a balloon. So the protein that we used is called C9. And so here's what you should really know. Germs use things that they have inside them to actually live. And so if you poke a hole and let everything loose, germs will die. So how do you stop things from becoming too fast? Or what do you do if things are going too slow, right? You don't want things to go out of control. You want them to be just right. So remember that sticky isn't always good. C3b can stick to anything, even your own cells. So what tells the macrophages that you shouldn't be eating the good cells and only the germ cells? They just want to eat. Well, one way is to actually stop more C3b from being created, right? Assume that this is the actual good cell that we have in our body. So if C3b big B were to attach onto it, right, before it could create more C3b, we just kick off that big B little b using this protein called the DAF, or the decay accelerating factor. And when you do that, although there's a C3b sitting there, it can't create more C3b, and so it's not as, tasty as a germ cell which has been covered with C3b by now. So how do you control it? Well, we have C3b on cells, but we don't want it to do its job anymore. So we use this protein called factor H. So factor H attaches to C3b, and it calls onto another one. It's like, hey, factor I, come here, cut this thing off. Factor I is like, okay. And so it cuts C3b into two parts, right? Now, C3b can't do what it usually does. It can bind to B and create C3b big B little b, and it can create more C3b, right? It can't do anything a C3b molecule would do. And so factor H actually stops it. However, this is still very, very tasty for macrophages. What if we combine both of these methods? Well, this is what the MCP molecule actually does. So the first thing it does, just like the DAF, it kicks off that big B little b molecule, and then it uses the I factor to cut the C3b into two pieces. So wait a minute, this is all the ways to actually slow down C3b, right? But what if we want more? Well, there's a protein called properdin, and we can make C3b big B little b an even better convertase if we attach properdin to it, and it starts making C3b really quickly. Well, what if the Mac is working too well? Remember that it's poking holes in things. We don't want holes everywhere, we just want them in bad germs. Well, there's three ways to stop it. One's called S protein, another one is called factor J, and the last one is called clusterin. These molecules stop C5b from calling for C8. Another way to stop C8 is to actually use protectin or HRF. So this stops C8 from attaching to C5b, C6, and C7 together. So what really happens when you get infected, right? What happens when you get a wound? Well, we call it inflammation. Let's see what it is. So when you hurt yourself, four things happen really, really quickly. Like, first thing is it hurts, right? If you fall or you trip or you get wounded, it's going to hurt. The second thing is your wound is going to swell up. Have you ever seen the bump that usually forms after you get hurt? That's what we call swelling. The third thing is your skin becomes really red where you got wounded. If you ever touched a wound, you'll actually notice that it's warmer than the rest of your body. This is what inflammation is these four things that happen when you get wounded. So inflammation is actually good for your body. It helps us to fight off the germs quickly. So what part of the complement system helps in inflammation? Do you remember those three proteins we made but never really used? They were called C3A, C4A, and C5A. They were created with most of our convertases, and we call those anaphylatoxins, because these proteins help to make inflammation better. So what do they really do? Well, anaphylatoxins help create more swelling, because swelling helps the wound heal quickly. They attract good cells from our body to the site of infection, or your inflamed site, where they can actually help to stop bad germs from doing their thing. Anaphylatoxins also go to these special cells called mast cells, right? And mast cells have these things called histamine. So histamine actually causes even more swelling. And so that way C3A, C4A, and C5A help create more inflammation. So let's do a quick review. What is the most important protein in the immune system that we find in the complement system? Well, it's C3b. C3b is a sticky protein that sticks to germs and helps us find it and kill it, right? C3b can be made through three pathways: the alternate pathway, the lectin pathway, or the classical pathway. Sometimes we have to make the pathways do more work, and we use things like Properdin to make it go faster. Sometimes we don't want it to go faster, and so we use methods of regulation. Thank you for listening to me, giving you this presentation. I hope you learned something new, and I hope you enjoyed. Thank you.